Hello students, I welcome you to EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, that is NDRI, Karnal in Haryana, India. Today, I am going to talk about simulating enzyme kinetics from the paper Biostatistics and Bioinformatics. Dear students, biochemistry is the study of chemical reactions catalyzed by enzymes produced by living cells of bacteria, plants, and animals. Enzymes are biological catalysts where they increase the rates of the reaction, that is, they speed up the reaction rate. They increase the velocity of the reactions. And this velocity of the reaction is increased in both the forward direction and the backward direction, because the reactions catalyzed by enzymes are technically reversible. Therefore, the enzyme catalyzed reactions achieve equilibrium concentrations after reaching at the equilibrium. The enzyme catalyzed reactions have the same equilibrium constant as the non-catalyzed reactions. That is, enzymes do not change the equilibrium constant. The equilibrium concentrations arrived will be same, but the equilibrium will be achieved very quickly because of the increase in the rate of the reaction. Therefore, after some time, products and substrate concentrations will not be changing after the equilibrium has been achieved. So this is represented by a kinetic parameter known as equilibrium constant. Each of the enzyme catalyzed reaction has been studied to develop this type of kinetic parameters. Another is Km, which represents the affinity of the enzyme. Numerically, it is equal to half Vmax. Vmax means velocity, which an enzyme can achieve. That's the maximum velocity an enzyme can achieve. When one molecule of an enzyme will not be able to have velocity more than Vmax. Similarly, we have other kinetic parameters such as allosteric Hill coefficient and say inhibition constants, many more. We'll see those. These parameters are combined together in the form of a mathematical equation to build what we call rate laws. These are the models which can be used for simulating reaction kinetics. These models are built from limited experimental data. This reduces the complexity of the necessary kinetic parameters so that we understand the actual reaction easily. Each of the enzyme catalyzed reaction which has a rate law with parameters in the rate law has associated data values for these kinetic parameters in the rate law. The enzyme, once an enzyme kinetic parameter containing rate law has been developed, these data values are studied experimentally and model is developed. And once the model and the parameter values are available, they are published in the research journals. These published values has been compiled manually by curators of a reaction kinetics database known as SABIO-RK, that is reaction kinetic database SABIO-RK. Another is Brenda. We'll have a look at the Brenda in the next module. In this module, we'll have a look at the SABIO-RK reaction kinetics database, and we'll be visiting and we'll be downloading data values as well as the rate law, that is adequate law model which has been developed to be used in simulation of the reaction enzyme catalyzed reaction kinetics in silico using a simulation software such as complex pathway simulator, commonly known as COPACI. And this can then be simulated reaction kinetics can be used to understand the time course, that with the passing of the time, how the substrate and the product concentration change, and at the equilibrium, the steady state level or the equilibrium level can be finally seen. Therefore, once the rate law and the associated values for the kinetic parameters in the rate law are available, these can be used for simulating reaction kinetics in silico. When we are able to simulate one reaction, then we can simulate the another reaction also. Say a next reaction in a pathway, for example, glycolysis, having 10 reactions from hexokinase to pyruvate kinase. So we can then enter another reaction for simulation, and after entering all the 10 reactions, it means we will be able to simulate complete biochemical pathway. Once we simulate the pathway, then we can vary the concentrations, initial concentrations of the various metabolites to study how the time course changes, how the values of the various substrate, the concentrations of the various substrates and the products changes. So therefore, this helps in manipulating the pathway in silico. We know that glycolysis shares 7 reactions with the gluconeogenesis pathway. Therefore, let's say that we are interested in studying glycolysis and gluconeogenesis in liver cell culture. Without actually performing the experiment, we can simulate and we can vary the concentrations of the various metabolites, initial concentrations or the enzyme expression levels, let's say. Therefore, this will help us to understand the pathway in silico before actually conducting the experiments. Therefore, the learning objectives in the present module includes understanding kinetic data of enzyme catalyzed reactions and enzyme kinetics databases for downloading kinetic data for simulating irreversible enzyme kinetics using downloaded enzyme kinetic data with the complex pathway simulator, that is COPACI software, and also to simulate the reversible enzyme kinetics using COPACI. Each of the enzyme catalyzed reaction has associated kinetic parameters such as velocity called rate of the reaction. It is defined as the amount of the product formed or amount of the substrate consumed per unit time. The factors affecting the rates of the reactions are the concentrations of the species involved in the reaction and the conditions such as pH, ionic strength and temperature. More the enzyme concentration, higher will be the rate. Similarly, substrate, concentration of the substrate has profound effects on the rates of the reaction. These are all used for studying the reaction kinetics. Similarly, product formed has also an effect on the rate of the reaction at a given time. Other ligands such as activators, which enhance the rate of the reaction, or the inhibitors which reduce the rate of the reactions, allosteric controls, all these are very important and we need to have the kinetic parameters for these species, concentrations as well as conditions. Thank you. The simplest enzyme catalyzed reaction in the cells is the one in which only a single reactant is present on each side of the reaction, that is, single substrate and single product. An example is provided by reaction catalyzed by glucose 6-phosphate isomerase, an enzyme involved in glycolysis in which glucose 6-phosphate and fructose 6-phosphates are interconverted. The Gibbs free energy change for this reaction is 0.6 kilocalorie per mole, which is very low, conveying that this reaction catalyzed by glucose 6-phosphate isomerase is a reversible reaction. Therefore, it can run in the reverse direction to produce glucose 6-phosphate from fructose 6-phosphate. Consequently, either glucose 6-phosphate or fructose 6-phosphate can act as a substrate depending upon the level of each and the metabolic needs of the cell. The reversible reaction sequence for glucose 6-phosphate isomerase can be represented using a reversible arrow for each reaction. A shorthand notation for writing down the kinetic mechanisms of enzyme catalyzed reactions has been developed by Cleland and has become quite popular. In this shorthand notation, the letters used for writing the substrates are A, B, C, D. In the order they enter or are added to the active site of the enzyme. Similarly, the letters used for products start with P, Q, R, S, and they are again used for the substrates in the order they enter into the active site of the enzyme. Stable enzyme forms are also represented using symbols starting with E, then F, G. The order in which they occur as stable forms during the enzyme catalyzes chemical reaction. The number of reactants and the products in the reactions are designated by the terms uni if there is only one reactant or one product substrate. Two substrates mean bi, mean two products bi. So if the reaction is having two substrates and two products, the reaction will be termed as bi in this shorthand notation developed by Cleland. Similarly, for three reactants involved, either substrate or products, the term used is ter, T-E-R, ter. And for four, quad, Q-U-A-D, quad. And for two substrates entering the active site, there may be the order which enters first, which enter later on, subsequently. Therefore, we have different notations for bi reaction. We'll have a look at that. In bi kinetic mechanisms, if the entry of the substrate and the leaving of the products occurs in an ordered manner, then this is known as ordered bi reaction mechanism, kinetic mechanism. In this case, the first substrate will be labeled or notated as A. A will enter E, and then this will produce EA complex. Then it will be followed by entry of the second substrate, that is B. Then this will form the complex EAB. This EAB will pass through the transition state to form EPQ. Now the first substrate product which will be leaving this complex will be termed as P, and the next product which leaves in the end is termed Q. So this is ordered bi kinetic mechanism. On the other hand, if the entry of the substrates and exit of the products is not in an ordered manner, that is, it is random, then this is known as random bi kinetic mechanism. In this mechanism, either A can enter in the active site first or B can enter. When A enters, it forms EA complex. When B enters, it forms EB complex. After the formation of either EA or EB, A will enter into the EB complex and B will enter into the A complex. With the formation of the EAB complex, EAB complex will then pass through the transition state to produce EPQ complex. Now, exit of the products is again random. That is, either P may leave first or Q may leave first. So, therefore, this is termed as, in Cleland shorthand notation, random bi kinetic mechanism. Sometimes what happens that the first substrate enters into the enzyme active site and forms EA complex, which passes through a transition state to form EP, and the first product leaves the active site. This is followed by modification of the enzyme, say an acyl intermediate, mean acyl group added to enzyme. In this case, the leaving of the P from the EP complex will leave behind not E but F, a modified form of the stable enzyme. Then the second substrate, that is B, will enter into this F, the modified enzyme, and form EB, which is then converted to EQ, and now Q leaves the product. Product Q leaves and gives the stable enzyme form, the original form E, the free enzyme. The symbols used in Cleland nomenclature for writing down kinetic mechanism are different from the symbols used in writing down reaction mechanism, which are used for developing enzyme rate law equations. The reversible reaction sequence for glucose 6-phosphate isomerase can be represented using symbols E for enzyme glucose 6-phosphate isomerase, S for substrate glucose 6-phosphate, and P for product fructose 6-phosphate, and ES for the enzyme substrate complex, and let us say EP for the enzyme product complex. This reaction involving a single substrate and a single product is called uni-uni reaction in Cleland nomenclature. The K1 shown here is the rate constant in the first reaction for the formation of enzyme substrate complex, that is ES complex, from enzyme E and substrate S. The K2 is the rate constant for the second reaction for the formation of the product from the enzyme substrate complex. K minus 2 is the rate constant for the third reaction which occurs in the backward direction, that is binding of the product with the enzyme to form enzyme substrate complex ES, and K minus 1 is the rate constant in the fourth reaction, that is dissociation of enzyme substrate complex ES to form free enzyme and substrate. When the breakdown of ES complex to EP is fast enough as compared to its back breakdown to E and S, then the reaction sequence is represented as irreversible reaction. Then the rate constant for this second irreversible reaction is represented as Kcat and sometimes as K2 in biochemical literature. The velocity V, or the rate of the overall reaction for the formation of one product from one substrate for an irreversible reaction, is given by Michaelis-Menten irreversible equation, where the velocity is given by the product of the Vmax and substrate concentration divided by the sum of the Km and substrate concentration. Vmax for the overall reaction is given by product of total molar concentration of the enzyme ET and the rate constants for the irreversible reaction for the formation of the product, that is Kcat. We will use this model to simulate time course of uni-uni irreversible reaction. Km is the Michaelis-Menten constant for the overall reaction and is given by the sum of K minus 1 and Kcat divided by K1, where K minus 1 and Kcat are the rate constants for the degradation of the ES complex, and K1 is the rate constant for the formation of the ES complex. Whenever we have to write down the Km, we will always write down the rate constants, that is the sum of the rate constant which are involved in the degradation of the ES complex in the numerator, and the rate constant which is for the formation of the ES complex in the denominator. The value of Km is of great importance. Km is the substrate concentration when Vmax of the reaction is 1/2, that is half Vmax. Therefore, it helps to adjust the substrate concentration to determine Vmax. For example, once we have an idea that okay, this is the substrate concentration, let us say 2 millimolar for the half Vmax, then we can adjust the substrate concentration higher than this to achieve Vmax, and the substrate lower than this to achieve the lower values of the initial velocities, so that we are able to plan the experiment. In addition, it is an indicator, that is Km is an indicator of apparent affinity of a substrate for an enzyme. If the value of the Km, numerical value of the Km is very high, then it shows that affinity is very low. A low numerical value of Km means high affinity. And conversely, therefore, a high numerical value of Km means low affinity of the enzyme for its substrate. The value of Km also indicates an approximate level of a substrate within the cell. High affinity means very low concentration of the substrate will be present within the cell. It helps in understanding even the regulation part also. In addition, Chem provides a means for comparing enzymes for medical importance of the same reaction, such as lactate dehydrogenase reaction from heart, muscle, liver for instance. Reversible enzyme catalyzed reaction involves 8 elementary steps. It includes binding of substrate with the free enzyme to form enzyme bound substrate and the dissociation of the bound substrate from the enzyme to yield free enzyme. Then the transformation of the substrate bound enzyme to form transition state bound enzyme complex, and in the reverse direction transformation of the transition state bound enzyme to substrate bound enzyme. This follows transformation of the transition state bound enzyme to form product bound enzyme, that is EP, and in the reverse direction transformation of the product bound enzyme to transition state bound enzyme. Finally, it includes diffusion of the product from the enzyme bound product EP complex to yield free enzyme, and in the reverse direction association of the product to form enzyme product complex. For this reversible reaction, let us represent ES complex, transition state complex and EP complex collectively with one symbol, that is ES. This is just for simplicity. In an alternative notation for rate constants are represented by combining reaction number and K1 for the forward and K2 for the reverse reaction. That is, rate constant in the first reaction in forward direction is thus represented with the symbol reaction underscore 1 dot K1. The reaction underscore 2 dot K1 is the rate constant for the second reaction in the forward direction. That is the formation of the product from the enzyme substrate complex. The reaction underscore 2 dot K2 is the rate constant for the second reaction in the backward direction. That is binding of the product with the enzyme to form enzyme substrate complex, ES. And finally, reaction underscore 1 dot K1 is the rate constant in the first reaction in backward direction. That is dissociation of the enzyme substrate complex, ES, to form free enzyme E and substrate S. The notation for the reaction number, that is reaction underscore 1 or reaction underscore 2, is always enclosed within brackets. The rate of the overall reaction for the formation of one product from one substrate for a reversible reaction is given by the Michaelis-Menten reversible equation. Let us expand the rate law equation, where lowercase letter v is the rate or the velocity of the overall reaction. Vf is the maximum rate of the reaction in the forward direction for the formation of the product, sometimes represented as Vmax F in biochemical literature. Then Vr is the maximum rate of the reaction in the reverse direction for the formation of the substrate, and sometimes represented as Vmax R in biochemical literature. Kms is the Michaelis-Menten rate constant for the substrate, represented as Km for substrate in the biochemical literature. And Kmp is the Michaelis-Menten rate constant Km for the product. Substrate is the molar concentration of the substrate, and product symbol used here is the molar concentration of the product. Vf and Vr is the maximum velocity which can be achieved at a given enzyme concentration, usually achieved at very large substrate and product concentrations respectively. The Vr, that is Vmax R, for the reverse reaction is given by the product of the total enzyme concentration, that is molar concentration, and the rate constant for the substrate formation. The Kms, that is the Km for the substrate, and this is given by the ratio between sum of the rate constants for the decomposition of the ES, that is reaction underscore 1.K2 plus reaction underscore 2.K1, and the rate constant for the formation of ES from the substrate, that is reaction underscore 1.K1. The KMP, that is Km for the product, and is given by the ratio between the sum of the rate constants for the decomposition of the ES, that is reaction underscore 1.K2 plus reaction underscore 2.K1, and the rate constant for the formation of the ES from the product, that is reaction underscore 2.K2. The ratio of the concentrations of the product to substrate at equilibrium, that is the equilibrium constant for the reaction, can be calculated using enzyme kinetic parameters following this Haldane equation. Therefore, we have two models for a uni-uni reaction, one for the enzymes following irreversible kinetics and other for the enzymes following reversible kinetics. We can use enzyme catalyzed reaction kinetic data values to verify which of these models is actually followed by the enzyme under experimental study. But before that, let us learn to simulate reaction kinetics using data from the literature or data downloaded from the reaction kinetics databases. Enzyme catalyzed reaction kinetic data values for various parameters, say for Km, Vmax, Ki, Kcat, etc. These are all stored in online databases and can be downloaded for simulation on the local machine. These can be downloaded in platform independent language known as Systems Biology Markup Language, that is SBML. SBML is a standard which is recognized internationally for the sharing of the reaction kinetics data, so that user can download the data from the different reaction kinetics databases in the format of SBML. SBML need not to be understood by the users. This is like HTML, hypertext markup language, means the language used to mark up the features. For example, in reaction kinetics databases, SBML currently is supporting marking of the 10 features, such as what are the species present in the enzyme, what are the parameters, others, conditions, all these, rate law. So these are the features. We'll have a look at that, all those 10 features. But at the moment, we need to understand only that the reaction kinetic databases available online provide support for downloading the model and the associated kinetic parameter data values in platform independent language known as SBML. In December 2015, Level 3 version 1 of the SBML supported marking 10 features of the biochemical processes, that is enzyme kinetics or even the biological processes, which we'll see in the next module, that is simulating or modeling biochemical pathways. So all these 10 features are marked using systems biology markup language. The 10 features of a biochemical process includes compartment, whether it is extracellular, membranous, cytoplasmic, etc. Second feature is species, whether the substrate, product, enzyme, activator or any other modifier. The third is the parameter, whether it is Vmax, mean kinetic parameter Vmax or Km or equilibrium constant, etc. Fourth feature is the unit definition for time, quantity and volume. Then the fifth is the rule, which is to be followed during simulation. Then sixth is the reaction, whether reversible or irreversible and following a particular reaction rate law such as simple Michaelis-Menten, allosteric activation or competitive inhibition, etc. And then the seventh feature is for marking the event, which is to be initiated during time course of a simulation, such as adding additional substrate or export of a product after achieving a specified concentration level, etc., with the support for including events with priority. And then is the function definition for individual reaction rate law to be followed during simulation. And ninth is the initial assignment of the levels, whether fixed or based on some mathematical formula. And finally, 10th feature is the constraint which is to be followed during simulation. The numerical values for various kinetic parameters are determined using experiments and they are published in the research journals. There are two online databases. First is SabioRK, second is Brenda. We'll have a look at the Brenda in the next module. These two databases, the curators present at these two databases, they extract the information or the data from the published literature and store in their databases so that these can be downloaded in platform independent language marked by SBML. The data can also be submitted directly to these databases by the experimenter from the laboratory experiments. SabioRK database include information about the biological sources, that is, and is not specific for a single organism. For example, let us say that we are having a pathway, say a glycolysis again, but among different organisms. Say lactic acid bacteria, lactobacillus, lactococcus, which are used for curdling of the milk, or even probiotics like pediococcus, enterococcus. Enterococcus is both pathogen as well as probiotic depending on the situations. But the question here is that let us say that we are having a pathway such as glycolysis. We want to model that pathway and we want to add each of the 10 reactions in the pathway, but we don't have the parameter values for all the 10 reactions from a single organism. Since these organisms are homologous to each other, they are having common family, they are descendant from a common ancestor. Therefore, all the 10 enzymes, even if studied in different enzymes, say first hexokinase in lactobacillus, then glucose 6-phosphate isomerase in lactococcus, and then phosphofructokinase from pediococcus and so on in different organisms, we can use these parameter values for modeling the complete pathway so as to understand how it will be behaving in the real cells, so that we are able to design the experiments for further. So in this way, it is to be mentioned here that the SabioRK is not specific for a particular organism. So it includes the data values from all the organisms, and then when we don't have all the enzymes from single organism, we can use from different organisms also. And data can be downloaded with the annotations in SBML and can be used for importing in a modeling software such as Copasi. At SabioRK, the database can be searched using keywords which may be including the name of the enzyme itself or name of the substrate, any other compound such as product, activator, inhibitor. So using advanced, in addition to adding keywords for the names of the species, either one can go for advanced search and combine the basic keywords with the Boolean, using the Boolean operators such as and, or, and not. For example, let us say that we are interested in the kinetic parameter values of the enzyme glucose 6-phosphate isomerase. We'll enter the keyword glucose 6-phosphate isomerase, and then we can enter the enzyme number. So we'll open the advanced search. In the advanced search, we'll choose and operator, that choose and enter the value, say for enzyme commission number, which is 5.3.1.9 for glucose 6-phosphate isomerase. So in this way, and you can again choose another and operator, further expanding advanced search using the organism, say Homo sapiens. So like that, we can have advanced search using search criteria at SabioRK. Even biological source such as name of the tissue or the organism, as we have seen, or even the kinetic data type or external identifiers such as KAG compound. KAG means Kyoto Encyclopedia of Genes and Genomes. Compounds are there, pathways are there. We can use the identifiers, KAG compound identifiers. In this way, this provides a very user-friendly and advanced searching system at the SABIO-RK database. Visit Enzyme database at Express server and search for glucose 6-phosphate isomerase. Click search button. The results are displayed. This shows that the glucose 6-phosphate isomerase has enzyme number 5.3.1.9. In addition, it has several alternative names. Visit SABIO-RK and click search menu to open search dialog box. In the search text box, start typing glucose -6-phosphate isomerase. The available entries will be suggested. Choose the one in which you are interested. And now open advanced search and select AND boolean operated and open drop down list to choose EC number and enter 5.3.1.9 as shown in the slide. Press enter or click add and search button. In the ensuing page, select AND operator again and open drop down list to choose organism field to search. In the adjacent text box, start entering Homo sapiens and click add and search button. The following result page will display 24 entries. Scroll down to see other entries. Only one entry could be shown in this slide. Now use filter options on the right top of the page. Click on the rate equation checkbox. This will update the search results automatically to display 14 entries. Now using filter options further to exclude mutant enzymes, just uncheck the mutant checkbox. This will again update the search results page to display 6 entries for only wild type enzymes. Click on the right arrow button of the first entry in the kinetic data column, that is the first column, to display kinetic data. The general information for the organism, tissue, enzyme commission, number, class, etc. and substrates, products and modifiers. Scroll down to find out about the enzyme protein data, kinetic law, parameter values, experimental condition and the published literature reference. The last section has information of the reference literature. Click on the PubMed hyperlink in the reference to download the literature of this enzyme. The Km for the enzyme is 445 micromolar and Vmax is 440. We'll use this data values to simulate enzyme kinetics. The PubMed page will present the abstract. Full text link is also provided along with abstract. Click on full text link to read the full text paper. Click on download PDF for this publication to be opened with Adobe Acrobat Reader if installed on your computer. This will open the target publications using PDF Reader installed on your computer. Read the paper to find out relevant kinetic data values and rate law. At the top of the SABIO-RK record for the current entry with entry ID 21894, check the checkbox on the top right as shown. A hyperlink for export, that is entries to export, will appear with a yellow background and this yellow background will fade very soon. Click hyperlink for entries to export to download the enzyme reaction kinetic data. In the pursuing page, click Write SBML button and save model in SBML on your disk. Downloaded enzyme catalyzed reactions saved in SBML can be simulated using simulation software such as COPASI, that is Complex Pathway Simulator. Visit COPASI, download COPASI, install COPASI in your local machine and then run it. COPASI has evolved from its previous version, GPASI, General Pathway Simulator. It has evolved and has very high capabilities, very nice capabilities. It has the capability for models representing how many compartments are there, how many species are there, how many reactions are there in the pathway. Even it includes the support to add events. For example, let us say during the reaction pathway or during the reaction you want to add additional substrate or you want to remove product. How it will simulate? Add, say after 10 minutes of the initiation of the reaction, you want to add additional substrate. Support is provided for adding these type of events also. Then it has the support for undertaking important tasks such as time course, that once the enzyme catalyzed reaction is simulated, what is the time course, how the substrate decreases and product forms. So in this way we can have the time course of the reaction catalyzed. Even interactive manipulations such as altering the initial concentrations is allowed using sliders in the COPASI. We'll have a look at that also. Steady state parameters is another task, that what will be the steady state values of the substrates and the concentrations after the reaction completes. It has a support for parameter estimation, means we have the data values for the enzyme catalyzed reaction. It can calculate the parameter values. If we input the data, it will tell you what will be the Km, Vmax, etc. It also has support for metabolic control analysis. So this is very advanced, and it has support for modeling 38 predefined models or the functions, mathematical functions such as Michaelis-Menten, irreversible, which we have studied, that is velocity, initial velocity is equal to the product of the Vmax and substrate concentration divided by the sum of the Km and substrate concentrations. Like that, 38 predefined functions involving reversible, irreversible Michaelis-Menten equations or allosteric inhibition, allosteric activation, by ping pong, all these mathematical functions are predefined in COPASI. And above all, COPASI is available free of cost, freely. One can download, install and use it for modeling biochemical processes or even enzyme catalyzed reaction or complete models of the pathways. Thank you. The COPASI window will open and it has 4 graphic user interface elements, that is 4 GUI elements. On the top is the menu bar having various menus. Below the menu bar is a toolbar for the frequently used commands. Below the toolbar, there are 2 panels. The left panel is the navigation tree containing nodes which can be expanded, and the right panel is the working space where various values can be entered. To simulate enzyme kinetics now, open the file menu and choose the command import SBML. To open the saved file containing glucose 6-phosphate isomerase enzyme kinetics data, which was downloaded from the SabioRK reaction kinetics database. This will display several warnings as shown. These warnings are displayed to convey that COPASI requires certain data values and parameter units which are not provided by the SBML file imported. However, just ignore these warnings at this stage and click OK. The opening working space panel displays the general information regarding units and literature for publication of the model. Expand all nodes in the navigation tree. The nodes can be expanded by the single click on the symbol of the node or by a double click on each of the name. So open model, then biochemical, then species, then reactions. Now set the quantity units to micromole in the working panel and click on React_1 in the navigation tree panel. And in the working panel, uncheck reversible checkbox and select Henry Michaelis-Menten irreversible in the rate law drop-down list. This will display working panel. Click the commit button on the left bottom corner of the working panel. This will change the Km and the Vmax to 0.1 by default. Therefore, double click 0.1 for Km and set this Km to 445. Similarly, double click 0.1 for V and set the value to 440, which we downloaded or which we read in the literature. Now click the commit button again. Now click the edit rate law button on the right top corner of the working panel. This will display the Michaelis-Menten equation, mathematical equation for irreversible kinetics. Double click parameter overview node in the navigation tree panel on the left side. This will display the values for initial species values as well as kinetic parameter values together. Set D-fructose 6-phosphate initial concentration to 0 micromole and click commit button at the left bottom of the working panel space. Expand output specification node in the navigation tree panel and select plots 0 by double click. Then followed by double clicking new plot in the right panel, that is working space panel. This will open new plot named as plot_1. Click on the new curve button. This will open a dialog box. Select model time under time node in the left panel to display model time on the x-axis. Now in the right panel under species node and transient concentrations subnode, select fructose 6-phosphate and click glucose 6-phosphate while the control key is pressed. This will select both the species to be displayed on the y-axis. Whenever we have to select more than one species, select the first species with a single click, followed by subsequent clicks for each species while the control key is pressed. This is used for all. Now click OK button. This will insert a new plot for display of the transient concentrations of the selected species on y-axis with model time, on x-axis of the plot_1. Increase the width to 5 for both the selected species. This will display broad, wide curves for both the species with the width of 5. Now open the tasks nose in the navigation tree panel and click time course. Set the time course parameter values in the working space as shown in this slide. Now click run button at the bottom left corner of the working panel. This will display the time course for irreversible conversion of glucose 6-phosphate to fructose 6-phosphate. This shows that within 6 seconds, all of the glucose 6-phosphate is converted to fructose 6-phosphate under Henry Michaelis-Menten irreversible kinetics rate law. Run capacity afresh, that is again, and expand model biochemical nodes and click reactions 0 node. Double click new reaction in the working panel. A new reaction will be added. This reaction will be automatically named as reaction_1. You can change the name. You can enter the name of the enzyme as the reaction name. The entering of a new reaction in the reaction text box in capacity will be clear as we look at the rules for entering a reaction in capacity. The name of the species, any species may be substrate, product, enzyme, activator, inhibitor, whatever the name. It is to be included within the double quotes. That is, when the name of a substrate, let us say glucose 6-phosphate, is to be entered into the reaction, then the name should be included within the double quotes. Otherwise, type the name of the substrate or the species continuously without any space in between, that is without any gap. Therefore, if you don't have a gap or a space in the name of the species, that can be entered without enclosing within double quotes. But when the name of any species, maybe substrate, product, enzyme, is having a space character, then it is to be enclosed within the double quotes. So therefore, make it a practice or a general rule that you will always enter the name of the species which is enclosed within the double quotes. Now, if there are more than one substrates, so enter the name of the first substrate enclosed within double quotes, then enter a space character and type plus sign to join the next substrate, second substrate. Again, the name of the second substrate is to be enclosed within double quotes. This is followed by again entering a space and the type of the reaction. I mean, you can continue entering more substrates if there are more than two also. So, once all the substrate has been joined together using plus sign, then you will enter the sign for the type of the reaction, whether the reaction is reversible in the forward and backward or it is irreversible reaction. The irreversible reaction is entered by typing a hyphen followed by greater than sign together without any space in between these two characters, but a space before a hyphen and a space after the greater than sign. This will convey to the Kopasi that the reaction you are going to model is an irreversible reaction. On the other hand, if the reaction is reversible, then use equal to sign. So, enter equal to sign and a space before equal to and a space after equal to. This will convey to the Kopasi to model the reaction as a reversible reaction. Then you enter the name of the first product and close within double quotes, a space, join other products, maybe 1, 2 using the plus sign. When all the products has been entered, enter a space and type semicolon. This will end the reaction. Then after this, you have to add the modifiers. For example, you can add the name of the enzyme. Simply again enclosing the name within double quotes if it contains a space. And then let us say there is allosteric activator. Add that modifier also here, but after the first modifier. Enter a space, add the name of the second modifier. Again enter a space, add the name of the third modifier and so on. So in this way, after all modifiers after products has been entered, this will be considered by the Kopasi for simulation. Enter the isomerization reaction between D-glucose 6-phosphate and D-fructose 6-phosphate by typing the character exactly for the name of the substrate and the name of the product. We entered the reaction as irreversible. Therefore, reversible checkbox below the reaction text box is presented as unchecked. This is detected by the Kopasi. To convert this reaction to reversible, simply check reversible checkbox manually. This will make the reaction reversible, and in the text box for the reaction, the symbol will change to equal to sign. Now open rate law drop-down list and select reversible mechanism maintain. Now click commit button at the bottom of the working space. The mathematical formula for the reversible mechanism maintain can be seen now by clicking edit rate law button. This will display the reversible mechanism maintain mathematical formula. The reversible mechanism maintain rate law is selected because the parameter values required for reversible mechanism maintain modeling are published in the table 1 of the reference literature which we have already downloaded. Now set the model parameters with unit for time in seconds, unit for quantity in micromole, and volume units in liter. Double click parameter overview node in navigation tree panel to display the values for initial species values and kinetic parameters. To change any value, click on the numerical value of any species or the parameter and enter the actual value, followed by pressing enter. Click commit button at the bottom of left corner of the working space panel. User is now ready to simulate glucose 6-phosphate isomerase catalyzed reaction following reversible Michaelis-Menten kinetic rate law. However, to display simulation results in graphics output, first enter the format for output. To display the time course of the reaction, expand output specification node in navigation tree panel and select plots 0 by double click, followed by double clicking new plot in the working space panel. This is the same as we did for irreversible reaction. This will open new plot _1. Click on new curve button. This will open a dialog box. Select model time under the time node in the left panel to display model time on x-axis. Now in the right panel under species node and transient concentration subnode, select fructose 6-phosphate and press Ctrl key and click glucose 6-phosphate. This will select both of these species to be displayed on y-axis in the plot. Click OK. Now open the tasks node in the navigation tree panel and click time course. Set the time course parameter values as shown in the working space panel here in this slide and click run button at the left bottom corner of the working panel. This will display the time course for the reversible equilibration between glucose 6-phosphate and fructose 6-phosphate as shown. This shows that equilibrium is attained at about 300 seconds after the initiation of the reaction following reversible Michaelis-Menten kinetics with the user-defined parameter values. Therefore, we can use enzyme kinetic data values to verify that which of these models is actually followed by the enzyme under experimental study. Dear students, in this module, we learned about the reversible and irreversible rate laws, that is Michaelis-Menten, and found that these two are different and which shall be followed for simulation is to be decided based on that which of these two laws is actually giving our experimental results. So, we have to simulate the reactions using Kapasi. We have learned to simulate reactions using Kapasi after downloading reaction data from the reaction kinetic databases such as Biochemistry, and we downloaded the data, kinetic data values in a platform independent language known as SBML, and then we imported it directly using the Kapasi software. At Biochemistry, all the data is actually compiled from the published literature. Even some data is submitted directly. And we have seen that at Biochemistry, we can have filtering of the data. That is, we want wild type enzymes or we want mutant enzymes like that. So we have seen that and we learned that which data we want to get. And then once we downloaded and saved in SBML, we imported using Kapasi and then simulated using both reversible and irreversible rate laws. And we found that when we are following rate law using irreversible kinetics, the complete or whole of the substrate was converted to product within 6 minutes or 6 seconds. And then when we followed the same reaction using Michaelis-Menten reversible rate law, we learned that after 300 seconds, the equilibrium concentrations of the substrate and the products are reached. Therefore, whichever of these two laws explains our actual experimental data obtained in the laboratory, we will be following that law to be used for modeling this reaction in the biochemical pathway, which we will be undertaking modeling biochemical pathways in the next module. I thank you all for visiting EPG Pathshala.